Good morning, guys. It's another Biology with Miss Cherry, and here we are today, recapping the insects and moving on to fish. So remind me from last lesson, how does an insect get oxygen? How do fish get oxygen? You might know, you might not. And which part of the fish is responsible for gas exchange? So pause and play again when you're ready, and we'll go through those answers. Alright, so an insect gets oxygen using its spiracles on the side of the abdomen. And if you remember, we had the spiracles, which are those gaps in the side, linked to the tracheae and then the tracheoles. And what we've got is the movement of gases in and out of those spiracles, resulting in a transfer of gases to regions of the body tissue. So if I show the flow of gas using some blue, we're going to get diffusion, movement of a high to low concentration from the atmosphere to the body tissues, down those tracheoles into the body tissue, and likewise carbon dioxide will come back. Now fish get oxygen from the water. So we're concentrating today on the properties of oxygen being able to dissolve. Remember, it's a charged molecule. There is a slightly polar side to it, and water is slightly polar as well, isn't it? If you remember when we've done the H2O lessons, we did the lesson on water. We've got a delta negative region on the oxygen and two delta positive regions on the hydrogen ions, or the hydrogen molecules in that water. And so for the final part, what part of the fish is responsible for gas exchange? You might know, you might not. But the part of the fish is the gills, not the gills. Okay, it's not a football team. Gills. There's little flaps on the side of the fish. If any of you have ever had a whole fish from the fishmongers, there's little flaps just behind the head. So today we're going to look at the structure of the gills. We're going to look at the adaptations of the gills, and then we're going to look at how the gills help take oxygen in. So today's lesson is 3.5. Gas exchange in fish. Now, you will notice that this is only lesson 0.3 on like YouTube or things like that. That's because we've missed out a couple of practicals. So we've missed out the surface area to volume ratio diffusion practical, but we've also missed out as well the required practical 5, which is a dissection of a locust to look at the gas exchange systems there as well. We will do it when we get back to school because it's quite an important one. But yeah, unfortunately, we're just going to have to skip those because I don't think asking you to source and cut up a locust at home is necessarily the fairest thing. So let's get into it then. Have a look. I've got a lovely photo here from an aquarium. I didn't take it. Don't at me. It's from the Googles. Have a look at the picture of the fish. What is their surface area to volume ratio like? Is it big? Is it small? Does that mean they can transport gases through diffusion, or do they need a specialised exchange system? And what are the properties of the scales covering them? So hopefully you've touched a fish, not like an actual alive fish, but maybe a dead fish, maybe a fillet of fish. This doesn't count if you've only eaten fish battered from a fish and chip shop though, so I'm talking about like fresh fish, maybe some sea bass. But yeah, pause, have a go answering these questions, and play again when you're ready. So fish have got quite a small surface area to volume ratio. You can see that all of their bodies are quite rounded. And normally they're also quite pointed as well. And this is normally to benefit them in terms of streamlining, so to make them more effective swimmers. But what that does do is that means that they are slightly rounder, which means that diffusion is not going to be a suitable way to transfer gases. And at the same time, that second question about the properties of scales, the scales are waterproof. It'd be really weird if fish were able to absorb water like sponges and just slowly expanded over the day. So because they're waterproof, we can't get gas exchange of those water-soluble gas particles, those gas molecules, things like oxygen, in and out of the body. Therefore, fish, like humans, like insects, have got that specialised internal gas exchange system that they need to survive. And this gas exchange system is very different to any of our other examples that we're going to look at over the next few weeks. So we've looked at insects, we're going to take a look at humans and the gas exchange system and the movement of gases around them, and then we're going to move on to plants. And fish are ever so slightly more complicated, but not because of the way that the process works. It's more just about the flow of things. But it's going to be fine, because we're smart, we're capable. Let's get into it. Now the gills are these little bits at the side of the body. As you can see, they look like little tongues poking through. But if I zoom in, you can see they've got lots and lots of layers. Okay, and that's visible also on this bottom picture. And the gills are the parts of the fish that are there for gas exchange. They're just behind the head, but we say that they're on the body of the fish. So they're sort of like where your neck would be. And each of those individual gill parts are called gill filaments. You have to be aware of the term gill filaments. It's an important concept when we're looking at how the gills actually work and the flow of water over them. You can draw the diagram if you fancy. Don't have to though, you're never going to be asked to draw them in an exam. And the gill filaments slot together like teeth on a comb, and they form a gill plate. Okay, so filament, and they layer together to form a plate. And I want you to think about what you know about maybe things like the villi, maybe things like the alveoli. How do you think having those filaments structured in that way makes a specialised exchange surface? So pause and play again when you're ready and we'll look at the answer. So naturally, your answer in your brain should have gone straight to an increase of the surface area. Alright, so if I've only got one gill or a gill plate that's not folded, that's not going to allow a lot of space for gas exchange. Conversely, if I've got this filament kind of structure that's layered together to form that plate, I've got loads of surface to exchange gases. So all the way around, each of those filaments provides an exchange surface. Really, really key, because it means that we can maximise the amount of oxygen taken in. So we're going to take a look in a second, but just as an advanced notice, I guess, fish transfer gases or exchange gases by water flowing over these. So if water flows over these gill filaments and they've got a massive surface area, that's going to help to increase the rate and things like that of the oxygen transfer and the gas exchange. We've also got the fact that gill lamellae, okay, so lots of stacks of those gill filaments. You can see here that we've got gill filaments all laid up together, forming a plate, but they're all slotted together to form something called gill lamellae. Okay, now these lamellae are found at right angles to the filaments. So if I've got my gill plate, as you can see in this diagram, it's called the gill arch, and then I've got gill filament, and then I've got lamellae, and lamellae is going to come up quite regularly over the next couple of years. It's generally just to do with the layering of something. So you can see here, these lamellae are offering a direction for the water to flow over. Don't worry about it too much yet. We're going to have a look at that in a second. But what these gill lamellae do is, again, they help to increase the surface area of the gills. So we're maximising the space that we've got for gas exchange here. Right, so we're using the perpendicular shape to show that we've got different areas or different directions that water can flow over and in turn move that oxygen around. So have a go at looking at this diagram. I just want you to make a couple of notes. How does oxygen, which is dissolved in water, get into the bloodstream of fish? Hey, look at the picture, maybe start over here and then work your way from left to right. Pause, spend a couple of minutes looking at this and play again when you are ready. So the first step is that fish are going to take in water through their mouth. That's that kind of like gub-gub mouth movement that you see fish doing. But the scientific word for their mouth is the buccal or buccal cavity. I don't really know how to say it. I say buccal, but it doesn't sound very, very good. I don't know, maybe you want to Google it. Okay, so they're going to take in that water via their buccal cavity, and what it's going to do is when they close their mouth, there's a change in pressure. Okay, so if you imagine I've got, if I take as much water into my mouth as possible and I leave my mouth open, that's all right. But as soon as I close my mouth, so closure of mouth, we're going to reduce the volume available for that water to stay in, and therefore we're going to increase the pressure of that buccal cavity area. Now what that does in the fish is it forces the water back. Okay, if you imagine we've got loads of water in our mouths, if I close my mouth again, that's going to force the water sort of backwards towards my throat. But in the fish, it moves that water over the gills. And because we've got that opening at the side of the head, that allows water to flow out. We get that pressure decrease. It's like a pressure reliever or taking the valve off of something. So the water is forced over the gills from the buccal cavity and removed into the water again surrounding the fish via those gill openings, the little flaps where the neck would be. At the gill lamellae, and this is really, really key, water flows in opposite directions to the blood flow. So we've said that we've got the gills in plates, we've got the filaments, and then we've got and this is going to be a horrible diagram again, I'll draw the lamellae in a different colour. We've got the lamellae that sort of stick up at right angles. Okay, as you can see in this diagram, we've got water containing oxygen maybe flowing from left to right, but the blood flow from deoxygenated, so let me just get a different pen colour, so deoxygenated to oxygenated has evolved to flow in the opposite direction. Okay, and this is a process called countercurrent flow. A bit like if you were to run, I don't know why you're running, if you were to walk downstream of a river while the river's flowing in the opposite direction, that's what's happening here. So imagine you're going to be the blood flow, but the water is flowing in the opposite direction to you. And this is helpful for a few reasons, and we're going to take a look at those in a second. But before then, have a go at this question. In relation to fish gills, okay, and that's the important bit, we're thinking about fish gills here, explain what is meant by countercurrent flow. And for the A level, you have to look at the context, because countercurrent flow also comes up in module 6 with the kidneys. So in this case, in relation to fish gills, explain what is meant by countercurrent flow. It is 1 mark, so pause now for a couple of minutes and have a go at writing a single statement sentence that summarises what is meant by it. Alright, so answers wise, it's not too difficult, and if you didn't get it, please take the time to write the statement out properly. The movement or the flow of water and blood are in opposite directions across the gill lamellae. It wouldn't have been enough if you'd have said that water and blood flow in opposite directions, because it specifically asked about the fish gills. So you had to mention the lamellae. That would be probably in the exam underlined. Okay, so don't forget our key terminology and what the context of our question is. Now, countercurrent flow is the pivotal part of gas uptake in fish. Okay, it enables constant and efficient movement of oxygen across those gill lamellae. And I'm going to be using this little diagram. If you wanted to, you can pause for a second and draw it out. I'm just going to make you aware that the dots are sort of oxygen molecules, oxygen concentration. So you can see on the diagram that there's a higher oxygen concentration where there's more dots and lower where there's less. Likewise, they've got a few numbers in there that are just there to help relate to the concentration. So throughout the next few slides, this is going to be my point of reference. There's going to be arrows and stuff. Again, you're welcome to annotate, or you can just write some notes down as we go through. But we're going to relate this to an exam question afterwards. So if you're more visual, maybe you want to spend a minute or so just drawing out this diagram. So we're going to start imagining that water is flowing from left to right. And in an exam situation, you can tell that the water is flowing from left to right because the left-hand side has got the highest concentration of oxygen. And that means that none has moved out of that water yet. So let's start looking at it from the left-hand side. Blood over here at the bottom where we've got lots of dots already has lots of oxygen. Okay? But it meets water that has got an even higher concentration of oxygen. Okay? It meets water that's got 100% oxygen in it. And we've still got a concentration gradient there. So from, if you imagine, even if we're just looking here, if we count the number of dots, we've got 3, 4, so we've got 10 dots there. So we can say 10 oxygens in the water. But in the gills, we've only got 9. So 9 O2 in gills. And therefore, using our knowledge from last module, we can say that there is a concentration gradient. You know, if we were to draw it, we've got 10 oxygens down to 9 oxygens. There's a concentration gradient. We can get that flow of oxygen. Diffusion will happen here. Yeah? Even at the time that the gills have got loads and loads of oxygen in them. At the other side, so on the right-hand side, we've got deoxygenated blood, because remember we said that we've got the flow of blood and water in opposite directions. So this blood is deoxygenated. It's got 0% or 0 molecules of oxygen in it. But at that point, the water has still got 1 molecule of oxygen in it. So again, we've still got a concentration gradient. No matter how small, it's still there, so diffusion can still take place. We've still got that concentration gradient, and the idea of maintaining a concentration gradient down the length of the gill is key. So this whole idea of countercurrent flow, and you can see maybe in this diagram a little bit better, means that oxygen can be exchanged continuously. And the key statement that you can get in the exam for a good answer is that the gas exchange system, the diffusion gradient, occurs across the entire width of the lamellae. Okay, and you might want to remember that this means that roughly 80% of the oxygen in the water is absorbed. And you can see in this diagram, probably a little bit easier than you could in the other ones, that there's always a higher concentration of oxygen in the water versus the blood, maintaining that concentration gradient. So now let's take a look at how it looks in an exam situation. We've been spending a lot of time looking at countercurrent flow in a diagram of fish, but sometimes they'll give it to you in this quite jank looking graph. Right? So in an exam, you might be asked to describe and explain the graph that you can see on the page. So have a go. Use the graph provided to describe and explain the countercurrent flow system in fish and how it is useful for gas exchange. So there's 2 parts to this question that we're going to take a look at. I'd like you to have a go at answering this before we take a look at how to do it properly. So the first bit is describe, so say what you see. And then it's an explain, so give reasons for. So give reasons for why you see what you can see. Okay, so we're describing the flow system, but then we're also adding a little bit of detail, so relate to biology, in how it's useful for gas exchange. So we should really have a point, an explanation, and an application. Yeah, so point, explanation, application. It's 4 marks, so you might actually want a couple of application points. So the first thing you're going to do is you're going to describe the graph, obviously, because that comes first in the question, then explain it, and then give me some reasons as to why it's useful. So pause now. I'd like you to spend 5 minutes writing your best answer, and then I will have a go at showing you what the answer should look like for maximum marks. So pause now, play again when you're ready. Before we take a look at what the marks should actually look like though, we're going to spend some time thinking about what you should have recognised from this diagram. And it's pretty standard, hopefully you will have got these points. These are not marks just yet. As the distance along the lamella increases, the percentage of oxygen concentration decreases. This is just a pattern that you hopefully recognised. So along the X we've got distance along the lamellae. You can see there that all the way down there is that negative correlation between oxygen saturation and distance along the lamella. In turn, or looking at the two lines, you might have recognised that the percentage oxygen saturation in the blood is always lower than that of the water. So there's no point, if I highlight, there's no point on this graph that this blood line, highlighted in red, is any higher than the water. So the amount of oxygen from this graph available from the water is always greater than that in the blood. So now we've discussed the patterns that you can see in that graph, let's take a look at the actual marks that you could get. So describe and explain, we're going to take each of those in turn. So my first point, the percentage of oxygen saturation in the blood is always lower than that of the water in the graph. And this is just something that we've discussed over the previous slide when we looked at the patterns visible from this graph itself. The explanation though, so our E point, this means that there is a constant diffusion gradient between the blood and the water. Remember we said that when we were looking at the patterns visible, the water is always higher in oxygen saturation than the blood. We've got point, example, or explanation, and let's do an application or an addition point. The constant gradient, so that constant difference, ensures that diffusion can happen across the entire length of the lamellae. Remember I told you that was the pivotal point. So far we've got 3 marks. We've described, we've explained, so that's our describe, explain is there. And then we've explained how that relates to gas exchange in fish, but we're not quite there yet. We've only got 3 out of 4 points. Final point is to say that this ensures that there's a constant diffusion gradient and allows up to around 80%, and you could have estimated from the y-axis of the graph that 80% of the oxygen is diffused into the blood. So if you didn't get that last mark, maybe you want to spend a couple of minutes now just sketching that graph and maybe highlighting where that 20% is. Make a note of it, it's important. It's not 100% transfer. It would be near impossible for biological things to be perfect, like 100% perfect, because there's always mutations and other factors that are going to affect this flow of oxygen and maybe the diffusion of oxygen as well. Now, just to make it a little bit more annoying, fish use something called countercurrent flow, but you also have to be able to recognise something called concurrent flow. So have a go. We'll go through the answers in a second, but I want you to use your brains for now. Use the graph and the diagram to explain why concurrent flow, okay, or parallel flow, as you can see in this diagram and here, why that would be inefficient for gas exchange. So look at how the diagram on the left with the little star differs from the diagram that I asked you to draw for the countercurrent flow. And explain why that would be bad for fish to use that to exchange gases with. So the first thing you should have recognised is that concurrent or parallel flow means that the water and blood flowing through the gills flow in the same direction. So same direction, blood and water. Okay. Now down the length of the gills, as you can see in this left-hand diagram, diffusion only happens until equilibrium is reached. So initially, yeah, cool, we've got lots of oxygen in the water transferring to the gills, but then a little bit further down, we've reduced the oxygen concentration in the water and increased it in the gills. And then eventually we get to this equilibrium position that means that there's about 50% oxygen saturation in both the water and the gills. Now this isn't great. Actually, if you're a fish, it's not going to be very helpful. So what we've actually got happening is that after that point, we don't get any more net movement of oxygen. So after that 50% saturation has been reached, there's no further flow of oxygen between our two areas. And you can see from the diagram on the right hand side that the concentration in both areas is the same. Diffusion is going to stop. That means that if you compare it to that countercurrent flow, the percentage oxygen saturation in the blood is only about 50%, which might not be enough to survive. Now let's take a look at some examples that they might use in an exam if they're feeling particularly nasty. And this is a sort of random pick a fish and give me some differences kind of question. So mackerel are fast moving, while plaice spend most of their lives on the seabed. So this is mackerel on the left, mackerel, and plaice on the right. There are differences in gill structures between these two types of fish. Suggest what the differences might be. Now suggest is use your knowledge. It's not expecting you to get it or to even know about the gill structures of plaice and mackerel, because I'm sure you've got better things to worry about. But using your knowledge and knowing that mackerel are fast moving but plaice are sort of quite sedentary, they just kind of float around with their weird face on the side of their head, but bodies facing the other way. What are the differences in their gill structures? So you might want to have suggested, being not really the correct term, you will have wanted to suggest that mackerel have more gill lamellae. They're fast moving, so they're going to need to increase the surface area of the gills, which means that we can get more oxygen exchange. They're going to need to exchange gases much more quickly because they're fast moving, but because they're fast moving, they also need more oxygen just to keep going with their respiration. Okay, so you remember that you need oxygen and glucose, give us carbon dioxide and water. And you can see there that if we run out of oxygen, regardless of if they've got lots of glucose, they're not going to make that energy that they need to keep being able to move. And the movement is key for the mackerel because it's a survival mechanism. They can dodge and move around predators. So now, obviously, we've reached the end. We're going to take a look at some exam questions. This is a very similar one to one that we started with. Describe and explain how the countercurrent system leads to efficient gas exchange across the gills of a fish. So pause, spend about 3 minutes, 3-mark answer, and play again when you're ready. For the second question, they've decided to bring in a random disease that you will have never heard of just to check what you know. So amoebic gill disease, or AGD, is caused by a parasite that lives on the gills of some species of fish. The disease, and I'm going to highlight the key terms here for you because this is a practice that I think you should probably be getting into. The disease causes the lamellae to become thicker and to fuse together. Right, so I haven't even looked at the question now, but I can recognise that they've told me that they're thicker and fused together. There's got to be a reason for that. AGD reduces the efficiency of gas exchange, so I'm going to highlight all of that. And the question is give 2 reasons why. So think about the key details that they've given you in that question. Why have they mentioned that the lamellae become thicker and how can that reduce the efficiency? And what about if the gill lamellae fuse together? Okay, give 2 reasons why, so pause and play again in about 3 minutes. And the final question that we're going to have a go at, woo la la. Table 2 shows some features of gas exchange of a fish at rest. The first row, we have got the volume of oxygen absorbed by the gills from each dm cubed of water in centimetres cubed. Then we've got the mass of fish in kilograms. And then finally, the amount of oxygen required by fish per centimetres cubed per kilogram per hour. And you have got to calculate the volume of water that would have to pass over the gills each hour to supply the oxygen required by the fish. Show you're working. You can do this quite simply, but you've got to think about it. So be careful. There's a few processes that you've got to go through. It's 2 marks. So pause and play again when you're ready. Oh, I lied. The volume of water passing over the gills increases if the temperature of the water increases. Suggest why. You're not expected to know, but perhaps you can use your knowledge from other modules to explain why this happens. So now we're going to take a look at the mark schemes. The first question was just asking about the whole countercurrent flow, explaining things. Water and blood flow in opposite directions. First mark. This maintains the concentration gradient along the whole length of the lamellae. And notice that the maintaining the concentration along the whole length are two separate marks. But for this question, I also want to draw your attention to this other marking point. Okay, accept a diagram if it's clearly annotated. So if you couldn't remember the actual words, but you could remember that diagram that had the flow, so you've labelled in the flows of water and the flows of blood, and they're going in opposite directions, and maybe you've labelled up some concentrations of oxygen, shown their diffusion, maybe you've sketched that graph that we've been through. That's fine, but it has to be annotated. Okay, now for concentration gradient, you need to have made sure that you have the idea that that gradient is always there, and that finally it's along the whole length of the gill. You could have said gill lamellae, you could have said gill filament, you could have also said gill plate. So they're quite lenient with that question, and hopefully you will have got 3 marks. For the second question, it was asking about that AGD, amoebic gill disease, I think it was. And it had those 2 different factors. So we've got thickened lamellae, but we've also got the fusion of the lamellae. And in each of these, you should have used your GCSE knowledge, if not anything else, to recognise that thicker lamellae gives a greater diffusion distance, and diffusion is underlined. It has to be in the answer. And the fusion of lamellae means that we've got a reduced surface area. Okay, you could have also said that you've got a reduced surface area to volume ratio, but the idea is that if you've got, if you imagine your hand, if I put my fingers all together rather than spreading them out, that's a reduced surface area, and that's effectively what that parasite is doing. Now, I'm going to show you how to do CII if you're not too sure. You should have got an answer of around 5.1 or 5.14. That's fine. And for CII, the question was asking about the volume of water and why it might increase the volume of water passing over the gills if it was slightly warmer, maybe. So you could have said that because it's slightly warmer, the fish might have an increased metabolism, maybe they've got an increased enzyme activity, so perhaps they're going to be opening and closing their mouths more so they can, take more water in anyway. But also, you might have recognised and remembered from when we did the water lesson that the higher the temperature, the less oxygen there is dissolved in the water. Okay, so higher temp equals lower oxygen concentration. And this is an important thing to remember, because if we've got less oxygen concentration in the blood, they're going to have to pass more water over the gills and the gill lamellae to ensure that that concentration of oxygen in the blood stays the same. Now let's have a look at CI, just in case you weren't too sure, and then we're done. So if you got that answer right for CI, perfect, well done. You can end the lesson here, but if you weren't too sure, just carry on watching. It's only a couple of more minutes. So the first thing that I do in any exam situation question like this is I have a look at the units that I've got and that I'm using, and also what the question is asking me. So it's calculating the volume of water that has to pass over the gills. Right. First thing I'm going to do is I'm going to see how much my fish weighs, because that's going to determine how much oxygen it needs. So fish is 0.4 kg or kilograms, and in each kilogram, or for each kilogram per hour, I know that they need 90 cm cubed of oxygen. So 90 cm cubed of oxygen per kilogram per hour. All right. So notice in the question, it's asking about the volume of water that passes over the gills each hour. So it's already like stepping us up to start answering this question using these numbers. So I've got 0.4 kilograms of fish at 90 centimeters cubed of oxygen per kilogram. So I'm going to start off by doing 0.4 times 90. And that tells me that I need 36 centimeters cubed of oxygen per hour for this fish. Okay, then I'm using the top value, the volume of oxygen absorbed by the gills from each decimeter cubed of water per centimeters cubed. And remember, this question is asking for the volume of water that has to pass over the gills. So now I'm using that top value. I know that there is 7 centimeters cubed of oxygen per dm cubed of water. Okay, so all I've done is I've changed the way that they've written those horrible rows into language that is nice and easy for me to understand. Now the final thing I've got to do is see how many dm cubed of water I need to get that amount of oxygen. Alright, so the final thing I'm going to do is I'm going to do 36 divided by 7, which gives me an answer of 5.142. Now, we don't generally like 3 decimal places. Try and keep it to 3 significant figures for your answer. That's normally good enough. So 5.14 dm cubed. Hope you found working through that useful. Let me know if you need anything. I'm always on Teams during these lessons, and I will see you again next time. Bye bye.